Welcome everybody to the Center for Evolution and Medicine seminar series. So I'm really excited to introduce today's speaker, Nick Banovich, who is coming home a little bit. So he did his undergraduate research in the lab of Center for Evolution and Medicine faculty, Dr. Ann Stone, and then went to do his PhD work with Yoav Gilad and Jonathan Pritchard at University of Chicago. And then in really spectacular form, negotiated a postdoc that transitioned to a assistant professor position at the TGen, Translational Genomics Research Center here in Phoenix. And maybe selfishly, I'm really excited to see Nick's presentation here. We've been talking about incorporating sex as a biological variable with another Center for Evolution and Medicine faculty, Dr. Ken Buteau. We've been rejected twice. In a week, in a matter of days, probably. But you can't get what you don't ask for, and I'm really excited about the projects that we're developing, and they're just a small bit of what Nick does. So I really hope you all enjoy his presentation today. Let's welcome Dr. Nick Banovich.
All right. Thanks for the awesome introduction, Melissa. She knew all those things about me. She didn't even have to look them up. She's a great collaborator. So, yeah, it's exciting to be back here. It's exciting to give a talk at ASU, and I am going to give a talk today that's like actually a little bit of a departure from sort of my normal work. I would say my core skill set, my core research interest is around how genetic variation alters gene expression and how those alterations in gene expression can lead to complex diseases or different complex traits. And this work is a little sort of off to the side of that. And this title here isn't even perfectly apt because I've added in some other new things at the end to try and hit the evolution part of the Center of Evolutionary Medicine. And but what's cool about being a speaker in front of you guys is that you are a captive audience and I get to do what I want. And so I'm going to take 1 minute here and just, reverse myself back and take a very brief trip down memory lane here. And so we even, there's sort of even a surprise audience member here who I didn't anticipate, which is Dr. Brenda Baker. So when I first came to Arizona State as an undergrad, I was in the School of Human Evolution and Social Change, and I got involved in Dr. Brenda Baker's lab. Was the first lab I worked in doing any sort of research. And if any of you know Dr. Baker, you know that means I was sitting in a room cleaning off pieces of skeletons from the Sudan, which was really exciting. And I was trying to figure out what I wanted to do and what my sort of core interest was going to be under this sort of anthropology umbrella. And I was interested in evolution, and I hadn't really thought much about genetics, and people kept saying, you need to take this course, ASM 345, human disease and evolution. You've got to take this course. And so, I was late to sign up to this course, but people told me I needed to sign up. So I went back, I dug up my old Yahoo email this morning and searched for Ann Stone and found this email, which is the first email I ever sent to Ann in August 13th of 2009, asking if there was going to be any extra spots in her human disease and evolution class. And ultimately, I just came and sat in the front row and just kept coming day after day until I think I broke her down and finally got her to agree to say okay. And I took this class, and anybody who's dealt with Ann knows that she's just a phenomenal teacher. And she gave me the opportunity to go do research in her lab, and that really catapulted me into the field of genetics. And if I hadn't had that experience, and if Ann hadn't allowed me to come work in her lab, you would have some other person up here talking about their junk today. So if my talk sucks, you can place the blame squarely on Ann Stone. So now I'll go into my actual work here. So I'm going to talk about two projects, and the first one is as what my title suggests, which is a project about multiple myeloma. So to give you guys a little primer on multiple myeloma, so I'm an assistant professor in the Integrated Cancer Genomics department at TGen, but I really know nothing about cancer. A year and a half ago, I was like, I need to figure out what's happening with cancer because I'm all of a sudden in this department, and I'm really a human genetics guy, and most of the things I do are relatively disease agnostic. So I've spent a lot of time trying to get caught up on some of these disease-specific details. So multiple myeloma is a disease of the bone marrow. Your plasma cells become cancerous, and it's usually due to translocations of the IgG and IgH locus to a number of genes that basically make your plasma cells start rapidly dividing and taking over all the space within your bone marrow. Now, this is a disease that has a very slow progression historically. People can have what's called smoldering myeloma, which kind of sits around without really rapidly taking over the bone marrow. There's a sort of pre-myeloma called MGUS that people can have for many years before it progresses into sort of true myeloma. And most people are diagnosed with myeloma sometime around in their 60s. And the median survival with treatment right now is somewhere around 4 to 5 years. And there's a 50% 5-year survival for myeloma. And it also doesn't affect a ton of people. So there's about 480,000 people worldwide who are affected by multiple myeloma. And because it mostly affects older people, because it's not incredibly aggressive, and because it's a sort of fairly small subset of the population, it's sort of historically gotten not a lot of attention. And this really changed with one sort of nonprofit organization called the Multiple Myeloma Research Foundation. A woman gathered up a bunch of people. She really wanted to find cures for multiple myeloma and put together a lot of consortiums and a lot of people in the same room to really start making advances in treatment. And at TGen, we've been involved with sequencing of this large clinical trial that they're running called the COMPASS study. And it's actually the largest sequencing study of any individual cancer there is. So we've sequenced, done whole genome sequencing, whole exome sequencing of tumors, and RNA sequencing of tumors from 1,000 patients, and we're following them over 5 years of their treatment. And this is a study that's led by Dr. Jonathan Keats at TGen, and so we've got a good sort of institutional background in myeloma. And because of that, we've ended up working with a number of people in this field, including some researchers at Mayo Clinic who were working with this novel treatment agent called LCL161. So this is one of these drugs that hasn't really come out on the market yet. It's still in clinical trials for a bunch of different types of cancer. But these researchers at Mayo put LCL161 into a mouse model of myeloma. And basically what this drug does is it inhibits cellular inhibitors of apoptosis. So there's these genes that get turned on that inhibit apoptosis, and they make cancer cells survive all sorts of treatments and become resistant to death. And this drug basically stops those genes from doing what they're supposed to do, either by releasing their binding partners or by degrading the proteins themselves. And it activates a whole bunch of things in the cell. It kind of acts like a transcription factor. It causes widespread gene expression changes in any, both to normal cells and tumor cells. And in myeloma, what's really interesting is, so like I said, it's in clinical trials for some other solid tumors, and in those solid tumors, it's directly killing the cell. So when you put this drug into the solid tumor, even if you do this in an in vitro experiment with tumor cells, those tumor cells die. But what our collaborators at Mayo found is that there's this really interesting mechanism of action in multiple myeloma, where instead of directly killing the myeloma cells, it's somehow activating macrophages and dendritic cells to target the myeloma cells. And so if you just put this drug into an in vitro experiment with myeloma cells, it shows no effect. They don't respond at all, they don't die, they just go along happily dividing. But if you mix in macrophages into this population, the macrophages start to kind of gobble up the tumors. But we don't really know what the mechanism of action is. We don't know why this is happening. We don't know what's being activated in the macrophages that's making them start to have this tumor sort of phagocytosis ability. And The reason we care about this is, like I said, there's a pretty dirty drug right now. It activates non-canonical NF-kappa B. It does a bunch of changes to the cells. And what we'd really like to understand is there a way that we could identify those gene expression changes that are specific to the macrophages, that we could try and develop drugs that would target those and specifically activate those pathways to then gobble up the tumor, rather than having to do kind of this complicated activate everything and it happens to work. And so we've become interested in a technology platform called single-cell RNA sequencing. So if you guys a little sort of step back on a primer on RNA sequencing, normally what we do is we take a tissue or a tube of cells, we grind up millions of them, we take the molecules of mRNA that come out of that, convert them to cDNA, and use next-generation sequencing to count the number of RNA molecules that were in that sort of mush, right? The problem is that it doesn't give us any information. It gives us an average picture of those however many cells we put in there. And so we don't get any information about what's happening in a cell type specific way. So what single-cell RNA sequencing allows us to do is, instead of taking this sort of gray blob and putting it in a tube without really knowing what's going on and sequencing everything, we can now sort of uncover and look at the gene expression patterns that are associated with these red cells or these blue cells or these green cells, and start to get this more high-resolution picture of the gene expression changes that are happening in the cell type specific way. So this technology is relatively recent, and it comes in a couple of different flavors. And each one kind of has pluses and minuses. So the first technology to really hit the scene was this Fluidigm. It's a microfluidic system. It's super expensive. It gives great quality data, but not on a lot of cells, and it takes basically an entire day to run one sample on this machine. There's some other assays that use, you basically sort cells into a 384 or 96 well plate and perform cDNA reactions within each well independently. And these also give fairly high resolution data, but again, it's on a limited number of cells, 96 or 384 cells at a time, depending on your plate setup. And they're also fairly time consuming. And then a few years ago, in 2015, this sort of new assay came out, which was they called it DropSeq, and basically what it does is it encapsulates each cell within a droplet of reagents within a mixture of oil, and then it performs the cDNA reaction within these droplets, and it actually puts a barcode onto each molecule of mRNA that's specific to that droplet. So then you process everything in bulk, and you're able to computationally on the back end deconvolute which molecules came from which droplet, because they got this little barcode address on each molecule. And so this is the technology platform that we're using. It allows you to profile up to 10,000 cells in parallel at one time, and it does it in minutes. So it takes only about 10 minutes to process through this machine to do the droplet encapsulation. And we use a commercialization of this called the 10x Genomics platform. And this is just sort of an overview, again, explaining what I just said, where you get your cells encapsulated in these droplets of reagents. The cells get this specific barcode that is specific to the droplet. And then one of the other things it does is it adds what's called a unique molecular identifier. So in addition to the little barcode that tells you which cell the mRNA molecule came from, it also, before any PCR, adds a second barcode. And what that barcode allows us to do is, after we do PCR amplification, we can tell which molecules were true unique molecules within the cell, and which molecules there's just a whole bunch of because by chance we PCR'd that molecule and I got 1,000 copies of the same molecule. And so I've just kind of illustrated that here, where you can see that, maybe there's 40 reads here, but there's actually only 4 unique molecules. And so instead of calling this 40 reads, we call it 4. And you can imagine situations where this weird PCR amplification bias could really mess up your actual calls, and you could think that there's much more mRNA within each cell than there is, and you could even get sort of biases where one cell looks like it has more mRNA, where in fact it only had a fewer number of true molecules. So what we did is we took a mouse model of multiple myeloma, and we performed single-cell RNA sequencing on this. And so here's the design. So this is a transplantable tumor. The tumor is grown up in a mouse, and tumor from that mouse is injected into other mice. And myeloma will colonize the bone marrow, and then it gives all of these mice end up with myeloma. And so what we did is we had two control conditions. We had a control condition where we didn't do anything to the mice. They were just totally normal. We had a control condition where we treated the mice that didn't have the tumor with the drug. We had 4 different conditions where we actually injected the tumor into these mice. So one where we gave them the tumor but didn't do any treatment. One where we gave them the tumor, we did this novel treatment, LCL-161. And then 2 mice where we gave them, or 2 conditions where we gave them the tumor and we treated with these 2 standard of care drugs. So cyclophosphamide and bortezomib are the sort of standard of care drugs in myeloma right now. And then the number inside each mouse is how many mice we had for each one of these conditions. So we had 6 control mice, 1 control treated with LCL, and so on. And ultimately we had 6 conditions, 18 mice, and this ended up in total, the experiment, we ended up profiling mRNA in over 92,000 individual cells. And we had about 17,000 genes detected on average. And so if you're sort of a bioinformatician, how many people are bioinformaticians in this room, just so I got a gauge? All right, a very small population. All right, so this is a big data matrix, right? So ultimately, if you think about this like an Excel sheet, we've got, an Excel sheet with 92,000 columns and 17,000 rows, right? And we have to figure out a way to take these data and make them interpretable and find signal in them. And so one of the things that's unique about this technology platform in a bad way is that it gives you a lot of missing data. So normally when we deal with data sets with traditional mRNA sequencing, I expect within each individual, I'm going to detect every gene, most of the genes in the genome. There's going to be very few genes that I have no reads coming from those cells that overlap that gene, right? With single-cell RNA sequencing, it's the opposite. Instead, what we get is, so here I'm plotting the proportion of genes with 0 counts, and this is across all of the cells. And so you can see that a huge number of cells have 90% of the genes don't have any reads covering them. So that means this data has a lot of zeros in it, which means that we have to think of sort of crafty ways to analyze it, because most of the out-of-the-box tools used for mRNA sequencing aren't ready to account for this type of missing data. So what do we do? Yeah. Yes, I will repeat the question.
Because you think there's a little dropout and you're not getting it, or because they just have those people turned on in those cells?
So I think so the question is this because there's a biological reason that these are off, or is it because of a technical reason? And the answer is, in this case, it's because of a technical reason. So what we sort of postulate is happening is that all of the steps that this 10x has to go through, or this library prep has to go through for the 10x genomics, it has to get encapsulated in this droplet. That little bead that's in there has a bunch of oligos that have poly-T that pull down the poly-A mRNA fragments, and then you have to do cDNA synthesis off of that bead, all within this very tiny reaction. That's an inefficient process. And so we get many molecules that actually exist in the cell that just never get converted to cDNA like they should. Now with other technology platforms like the plate-based and the fluidigm, the microfluidics-based, they tend to have far less dropout, and so that's when we start to get into more of the questions of biological sort of dropout, where you just don't have a gene on in one cell and you do in the other. But here we really expect most of it is because of technical issues. So the way we handle this is actually similar to how we handle a lot of big data genomics problems is we do a dimensionality reduction procedure. So basically the idea is instead of trying to look at the variation within each gene, we try and pull out these major components of variation across the system. And so this can be driven by many genes that are all acting in parallel. So you could imagine a network of genes that were all sort of upregulating or downregulating in the same direction would be one sort of major source of variation. And we do these dimensionality reduction methods. So some of you are probably familiar with something like principal component analysis, which is a linear dimensionality reduction method, which means that it can only find things in a straight line. And then there's other ones that people use, like this TSNE, which is T-stochastic neighbor embedding. And the reason people use this is because it can find nonlinear relationships in your data. So for example, if this is what my data looked like and I applied a principal component analysis, it's going to only be able to draw straight lines through this data, whereas TSNE can actually say this is the important component of variation. And so what you end up with is sort of this reduced representation of your large matrix. And you end up with a much smaller, much more manageable matrix that's got values that correspond to the major sources of variation in your data. And so we use these in different ways. And so generally the analysis that we're working off of, the principal component analysis is the workhorse that goes into all of the sort of underlying analyses. And the TSNE plot we use for visualizations. And the reason we use it for visualizations is because it looks really cool. And it makes very nice like clusterings of cells when we plot them in two-dimensional space that are easy for us sort of biologists to interpret. So I'm going to show you a picture here of a visualization of the data. So this is a TSNE plot. I'm going to show you a lot of these. So you can think of it like this. The x-axis is the axis that explains the most variation in this data. And the y-axis is the second most variation of the data. And this is a two-dimensional representation of these data. But there's actually many, many components of this that we could look at. So we could look at the fourth component, or the fifth component, or the 200th component of this data. But these are the two major sources of variation. And so it just allows us to take all the variation in this data and try and put it in a 2D space. So here, each dot represents a cell. And so this is you have to think that this clustering is based off of the total transcriptional profile of the cell. So when you see things that are close together, it means that these have more similar transcriptional profiles. And so what we're trying to do here is basically what I showed you in that first picture. We're trying to make that gray blob into something where we can identify specific distinct cell types within our data set. And so we can start to look at this in different ways. So first, I've got good metadata. I know what the different treatments were in this. So I can now pseudo-color these by what are the different treatment conditions. And I can see here's a big blob that's kind of specific to my cells after I give them the tumor. Here's a big area where it looks like is mostly cells coming from the untreated conditions. And then what we can do is we can try and sort these out into specific groupings. And the way we do that is using a graph-based clustering. And so I'm not going to get into the minutia of this. It's fairly complicated, but the general idea of what we're trying to do is if you imagine each one of these letters is a cell, we're trying to find sort of neighborhoods of cells that are all in the same region together. And then we use a cutoff to say, okay, I think this is one group. I think this is one group. And you can imagine we can change what those thresholds are. So I could have them be more restrictive, and maybe I separate these two into two separate clusters, or I could have them be less restrictive, where maybe it would group these now together all into one cluster. And so we've done a bit of data processing to this point. So before I really jump into the results, I just want to walk you through what we've done. So we took the RNA, the single cell RNA-seq off of the sequencer. We mapped it to the genome. we took those large numbers of counts and we collapsed them down based off of these UMIs. So now we're dealing what we would consider sort of true molecules of mRNA that were originally in the cell. We do some cleanup based off of missing data or really bad cells that didn't work well. We do some normalization, the dimensionality reduction, and then we just did this clustering I talked about. So here's a picture now where I've pseudo-colored it by this clustering. And remember, this is unsupervised. So it is grouping these cells together based off transcriptional similarity without input from me on what these things are. And so it's sort of defined 10 groups of cells. And what we then are able to do is go through and start to do some post-hoc cell type assignment. So we look at the expression of canonical marker genes, and we can say, all right, I think this cluster is made up of T cells. In this example, I'm showing CD3, CD4, and CD8, which are 3 canonical markers of T cells, and you can see that they all light up in this sort of purple population, and so I've called these T cells. And we do this for each of the different cell types, and here I've broken it down just into 6 major cell types. We've got B cells, erythrocyte precursors, monocytes, the myeloma cells, natural killer cells, and T cells. So then what we're able to do is start to do analyses in the sort of cell type specific way. And so the first thing we wanted to do is just say, does the single cell RNA sequencing at the highest level recapitulate what we see experimentally? And so the easy thing is to say, are there, in the cases where we treat with LCL161 that really has great tumor killing ability, do we see a reduction in tumor cells? And the answer is yes. You can see that when we leave the cells untreated, about 61% of the cells from those mice are tumor cells. 54 when we treat with bortezomib. It's actually slightly higher when we treat with cyclophosphamide, which is maybe bad news. And then we see a pretty drastic reduction when we treat with this novel agent. So this is what we observe experimentally. It's what we observe by microscopy, and it's recapitulated by this technology. But then what we're really interested in is drilling down on this monocyte myeloid population where the macrophages and dendritic cells live, because this is this sort of interesting population that's going through and somehow gobbling up all of these tumors. And so what we do is all these green cells here, and again, remember, the sort of green clusters look a little bit far apart on this plot, but this is, again, only a 2D representation of these data. When we perform these clusterings, they're actually done on the top 20 dimensions of the data. So even though they're a little bit spaced out in 2D space, they actually have really similar transcriptional profiles. So we take out all of the green cells, and we sort of run through the whole processing procedure again. Because when you think about identifying the major components of variation, if you've got 10 different cell types, that's going to be the thing that's separating out your data. The difference between a T cell and a B cell is the thing that is really making this data different. But now that we've drilled down on the small subpopulation of cells, we want to see what are the things that are separating out this particular myeloid population. So we reanalyze, and now these green cells kind of pop out into this new TSNE plot that looks, like another blob in space, right? But we can do the same sort of clustering analysis, and here we have identified 6 clusters, and then we look for this LYZ2 gene, which is a marker of macrophages, and we can see that there's kind of these 2 areas up here that are lighting up with this macrophage marker, and through some other canonical markers of macrophages, we became pretty convinced that this sort of light peach is the major macrophage population in our sample. So now we're able to go in and look at what's different between our treatment conditions. And so here's where we got sort of excited, right? So one of the things that our collaborators found was that you needed both the presence of macrophages and the presence of the tumor for the macrophages to start having their killing ability. So what they did is if you treated the macrophages in vitro separately and put them in with tumor cells, they do not start phagocytosing the tumor cells. If you put the treatment in with the tumor cells alone, they don't die, nothing happens. But if you put the treatment in with the tumor cells, take the supernatant from those tumor cells, put it in with macrophages, and then put those macrophages back in with the tumor cells, they go to town and start gobbling things up. And so this is why we had this condition where we treated a mouse without the tumor and we treated mice with the tumor, because we really wanted to see what's specific to those macrophages that both, or those mice, the macrophages in those mice that both have the tumor and have been treated with the drug, compared to the macrophages from mice that have just been given the drug but not the tumor. And so I've pseudo-colored these here. On the left, I've broken just these two populations out. So the LCL161 wild type are from the mice where they did not have the tumor, and the LCL161 tumor are the mice that did have the tumor. You can see that there's kind of a concentration in a few different spots, but particularly in this macrophage population, there's a little concentration there. All right, so here's the sort of exciting results if you like single-cell RNA-seq like I do. Here's the things I get excited about. So we went through and we performed a differential expression analysis. And we use a technique that is specifically developed for single-cell RNA sequencing. And so it models the fact that there's so many zeros in the data. And what we found are these number of genes, and this is 6, there were about 15 of them in total, that show almost exclusive expression in macrophages or macrophages and some of these other sort of myeloid populations from the mice that both had the tumor and were treated with LCL161. And so only when both of those conditions are met do you see these gene expression signatures. And so we think that these are probably the expression signatures that are really underlying this, this phagocytosis process. And I know it's a little hard to appreciate on this plot because it's hard to sort of visually match these two things up. So I've also shown here this really sort of poorly crafted box plot because the whiskers for some reason go up to the top of the cells. But this is a box plot, so the majority of the data here is at 0. And this is actually now, these box plots weren't generated from just the myeloid monocyte population. This is from the entire 92,000 cells, okay? So across all 92,000 cells, you can see that it's only expressed in the small subset of cells. And all of those cells are these cells that you see lit up in the box. So that means this is less than 0.01% of the cells are exhibiting this marker. And so what that means for us is that you can only really identify the signal using something like single-cell RNA sequencing. Because if you looked at this analysis in bulk, that gene expression pattern is washed out because it's such a small proportion of the cells. And so we're still doing some work to try and build some sort of do some kind of pathway analysis and network analysis of these genes. And then starting to move forward, our plan is to do functional validations using things like CRISPR-Cas9 and then other sort of knockout mice to see if we can induce or repress this response sort of in vitro and in vivo. So in summary for this kind of part of the talk, we think single-cell RNA sequencing provides a really powerful tool to study these tumor immune reactions in multiple myeloma or any kind of cancer we might be interested in. And that this analysis of individual cell types allows us to get information that just wasn't really possible from a bulk RNA sequencing perspective. And then we're really excited that we were able to identify these very exclusive and specific gene expression signatures and macrophages that we're hoping to build upon and might have some ability to lead to novel treatments for myeloma. Okay. Carlo, get it, yeah.
Thanks. And you'll have to repeat this in the recording. If you sorted the macrophages and did the RNA-seq on the bulk sort, would you see it, or are they still too rare?
You know, it's almost like you're a plant. This is like such a great question. So the question is, if we sorted these macrophages and just did this RNA-seq experiment, would we still see the signature? So the answer is yes, we would see it, but without the other cells to contrast, there's so many gene expression changes that happen when we treat with LCL161. I'm only limiting myself now to these ones that are very specific when I have this nice contrast both with the other macrophages and other treatment conditions and all of the other cells in the population. The vast majority of gene expression differences we see are shared across all of the cells. So when I look at this in bulk, or when I look at this in bulk macrophages, we'd identify thousands of genes with differential expression, and most of those would be the same signals that are shared across all or many cell types. Okay, so now I'm going to shift gears just completely and talk about something that's just incredibly depressing. And so, everybody get ready to feel really sad. And just everybody be really glad that I spared you the pictures, which is what I've had to see for the past few years. But I'm going to talk about transplacental cancer. So there's, in very rare occurrences, a mother who has cancer and gets pregnant can actually transmit that cancer to the fetus or to the placenta. And it's an incredibly rare occurrence. So in a literature search from 1866 to 2003, it was only found 87 times that this happened. And most of the times it results in fetal death, that it's just not compatible with life because the cancer takes over so rapidly that it outcompetes the fetus. And even sort of, it can happen both the placenta or to the placenta and fetus, and it's even sort of rare if we only look at these times where it actually metastasizes both to the placenta, or where it metastasizes to the fetus. So in 2011, TGen got contacted by Phoenix Children's Hospital because a case of transplacental melanoma had occurred. And so there was a woman who in 2006 had melanoma had been. So the other problem is I deal in both melanoma and myeloma, and they sound very similar. And so I work very hard not to say multiple melanoma, but you might catch me slipping up since I just spent 20 minutes talking about myeloma. So there was a woman. She, in 2006, had been diagnosed with melanoma. It was a small primary melanoma on her head. She had it removed. It was good margins. There were no complications, and she went on with life. She became pregnant, and in 2011, she gave birth to a healthy girl, and then just a few months later, she got diagnosed with incredibly advanced stage 4 melanoma after she had a number of seizures and other problems. Shortly after that, the infant developed 2 dark spots on her scalp, and they took her into the doctor, and it turned out that she was also had widespread metastatic melanoma, and that this melanoma had been transmitted from the mother to the infant. So if melanoma is one of these diseases that it mutates very quickly, so the cancer is often very drug resistant, but there is a kind of canonical mutation in the BRAF gene, the BRAF V600E mutation, that's targeted by a really good single agent therapy that has low side effects and really just sort of makes tumors melt away. Unfortunately, with most people, that response isn't very durable. It lasts for a few months, and then the tumors come back, and it's ultimately resistant to the treatment. But this drug had never been this tumor happened to have this mutation, but this drug had never been given to an infant before, obviously. And so Phoenix Children's Hospital worked together with the manufacturer of this drug to come up with a dose for the infant, and they were able to treat the infant, which actually had very good response to the drug, and the mother unfortunately did not have a very durable response. She ultimately passed away, and then the infant ultimately passed away because there was one intracranial metastasis where the drug doesn't cross the blood-brain barrier very well, and they couldn't really get a handle on the intracranial metastasis, and the infant died from surgery complications and complications from trying to figure out how to get rid of that metastasis. So, super depressing, right? But it is sort of an interesting case that we were able to collect samples from both the mother and the infant and do sort of the first genomic profiling of a case of transplacental cancer. And so we were able to collect 2 lesions from the mother and 2 lesions from the infant. And ultimately, we actually just found out about a month ago that the intracranial metastasis they still had samples of at Phoenix Children's Hospital. And so we just performed exome sequencing on that sample as well, although we're still, we haven't gotten the results back yet. But we had a brain sample from the mother, a shoulder sample from the mother, and then 2 samples from distinct regions on the scalp of the infant. And then we also got a germline sample from the mother. We got a buccal swab from the mother. And then ultimately, at the end, when I got involved in this project, obviously this was started in around 2012 while I was, just barely out of Anne's lab. And when I got involved in the project, it became really clear to me that there was many mutations in the infant sample that we were calling sort of subclones of the tumor that were actually contaminating infant cells. And so they're actually germline mutations from the father that look a lot like sort of subclonal mutation in cancer. Because remember, the cancer is fully the mother, right? So the cancer should have the same germline as the mother, but there were we're not getting perfectly clean dissections, and so we get these infant samples as well. And so we got a blood sample from the father that we used to sort of mask the germline father mutations. So, for some reason these have an animation. I'm just gonna, there we go. All right, so these were generally pretty poor quality samples. If you deal with sort of weird cases or clinical cases, you don't have much choice, right? You have to deal with what samples you can get. And so this is an example. This is actually a picture of one of the samples, one of the biopsies. It was 99% necrotic. And you can see, it was a half centimeter in length. And so we were able to still, isolate cells from that and perform whole exome sequencing on all of these samples. And so what we were able to do is build sort of a general picture of what was happening. We know that this melanoma started on the mother's forehead. And then we were able to find these sort of trunk mutations that were shared across all 4 lesions. We were able to find a number of mutations that were specific to the shoulder, in particular this BRAF L567V, which was the mutation that made the tumor resistant to the single agent treatment and was ultimately the tumor that continued to spread and resulted in the death of the mother. And then a couple of unique mutations to the infant samples, and the brain lesion actually had what we call a mutator phenotype, so there were hundreds and hundreds of mutations that were unique to the brain because it just was sort of inducing many mutations. So we got interested in this, though, from kind of an evolutionary perspective. And we thought this is kind of a unique case to think about tumor evolution. And so you guys might have seen, if you thought much about cancer, and in cancer, Carlo has a half smile here at a picture like this, but there's these sort of different models that people think about how cancer evolves. And there's kind of this linear evolution where there's a mutation in some proportion in one of the cells, and then that mutation grows to fixation and kind of becomes the dominant only clone in the tumor, and then another one arises, and then it gives birth to a new sort of dominance, and this is as you're sort of going along the life history of a tumor. And then there's this idea, which I think is sort of maybe becoming more generally accepted in the field of this branching evolution, where you get some subset of the cells get a mutation and they kind of expand and they form a subclonal population, so that within any given tumor, you don't have one sort of representation of that cancer. You've got many sort of subclonal populations that each have their own sort of suite of mutations. And this is kind of consistent with the idea soft, for the purposes of treatment resistance, I think about this as like the ability for soft sweeps to occur, right? That there's a lot of standing variation that exists within the tumor population, and when I give a treatment, there's probably already variation there that makes that tumor resistant to treatment. And then when I perform a treatment, that those alleles rise in frequency, but they already existed. And so we used a joint sort of analysis technique that was developed at TGen by a now research assistant professor, Rebecca Halprin, that sort of jointly models across all of the samples and tries to pull out these subclones. And we indeed find that, so this plot's really messy. I don't want you guys to try and fully dig too deep into it, but what I really want you to pay attention to are these plots on the right-hand side here. And these are plots that are representing the different sort of major clones within the samples. And so it's trying to jointly crawl them across 4 samples, but what you can see is that this brown represents the germline. So everybody has the germline mutations of the mother. That makes sense. And then you've got these different subclones that are at different frequencies within each tumor. So first we see intra-tumor variation, where within each tumor there's multiple subclones. And then we see inter-tumor variation, where between the tumors you see these different subclones sort of rising and falling in frequency. And this is the most clearly evident in the brain metastasis, where if you remember I told you there's this mutator phenotype that was specific to the brain. And so you can see that's represented here by this blue bar, which represents all of the variants in that kind of mutator phenotype, and that never shows up in any of the other tumor lesions. So we do see this intra and inter-tumor heterogeneity kind of consistent with this branching evolution model. But what I really started to get interested in is that what I think is particularly unique about this case is that when I'm thinking about, there's very little known about sort of placental transmission of cells. And me and Melissa have spent a bit of time talking about this, but the idea of there's probably always some cells from the mother that are transmitting to the infant, and some cells from the infant that are being transmitted to the mother. And I think there's good work to support that. And if you happen to have cancer while you're pregnant, there's some minuscule probability that the cells that transmit to across the placenta will be cancer cells. But I have to imagine that this is a very small, if not single cell that passes, a very small cluster of cells. And so my hypothesis is that there's not a lot of heterogeneity crossing this placental barrier. So even though there's a lot of heterogeneity both within the individual metastases and sort of across the mother as a whole, that I think there's probably not a lot of heterogeneity going into the infant, and that this was probably a very homogeneous disease entering the infant. And so the first thing I did to try and quantify this was just do a very sort of simple Spearman correlation, where I took all of the variant calls that we're very confident in. So this means I didn't say it was a variant in the mother, and then it was just really low coverage, and so I called it crappy in the infant. This means I had to have a strong yes or a strong no either way. And I just did a Pearson correlation for presence and or a Spearman correlation for presence and absence of these variants. And what I see really quickly is that the 2 lesions from the infant, so in this pseudo-coloring, purple is a higher correlation and green is a lower correlation. So it became immediately clear that the 2 infant lesions are much more similar than the other lesions. And I'm hoping that our third sample, which will be from this intracranial metastasis, will also share this pattern. Because even though they're both in the head region, the distance between sort of a scalp metastasis and an intracranial metastasis is about as far away as you can get. And the infant lesions are more closely related to the shoulder metastasis, which also makes sense, and then that brain metastasis is kind of off on its own because of that mutator phenotype. So we also wanted to quantify this in like a more clever way, and so we used, there's a package called Triomics, and basically what it tries to do is use Bayesian inference to construct phylogenetic trees of the samples. And basically, it's putting a prior on if it sees a mutation once, it never expects that you gain a mutation, then lose a mutation, then gain a mutation again. And so it's trying to build a phylogenetic tree sort of off of those principles, which I think is pretty well grasped by most of the people in this room. Normally, this is the thing I have to explain to people, right? Not the single-cell RNA sequencing. So it's the inverse of my normal talk. But what we see is that, again, the two infant lesions are very similar to one another. And we've got this sort of different measures of similarity, either using Jacquard distance, which is just a measure of similarity between the samples, similar to Euclidean distance in some respects. And then this metric of genetic distance that the program calculates. And you can see by either metric, the two infant lesions are really close to one another. But what we got excited about is that we almost have a molecular clock for the mutation rate of these samples. Because I know this at max, if this tumor transmitted at day 1 of conception, this infant was diagnosed with these sites were sampled when the infant was 2 months old. So at most I've got 11 months between time of transmission to time of collection. Probably less. It's unlikely this was at day 1 of gestation, right? But one lesion acquired, so just looking at the differences between these two sites, I now know that in this, let's say 6 to 11 month period, that these two tumors acquired 11 and sort of 30 novel mutations each, which kind of gives me a calibration for the mutation rate of these tumors. Because one of the things that, if we think about the fact that these were collected in 2011, the original melanoma was removed in 2006 and likely developed, the sort of initial mutations likely developed even some years prior to that. So there's a long history of this tumor that we're just really unable to look at this life history of a tumor normally, because sometimes they often sit in people. I've seen estimates of 20 years that, from the sort of initial cell acquiring that first sort of cancerous mutation. Whereas here, we've now got this homogeneous disease that went into the infant, and I'm sort of really able to look at the mutation rate of this cancer over time. And so when we started thinking about this, we want to kind of look at this hypothesis from a second direction. And these results are still relatively new, and I'm trying to get my collaborator to give them to me in a form that I can actually do some statistics on them. But basically, we hypothesized that if there was this reduction in heterogeneity going into the infant, that those tumors across the infant, the different metastases, should respond more homogeneously to the treatment. And so it's a little bit difficult to appreciate by these plots. And this is a really grainy, terrible plot, which looked much better on my computer. But the idea is that this is a metric of tumor size. It's actually a metric of tumor metabolism, but you can think of it as a metric of tumor size. And this is the baseline on treatment. They call it progression, even though it wasn't. This is sort of off. The infant never went off treatment. And so it's just sort of a progression for the mother, but continued treatment for the infant. And what you can see is that this infant continued to have response to the treatment. And what you can see sort of visually, and what the clinicians agree, and what the sort of data I'm actually trying to get, is you do see a much more consistent effect across the metastases in the infant, where really the only metastasis that doesn't respond is the intracranial metastasis, which continues to grow. But you can see in the mother, you've got some that get smaller and go away. You've got some like this lesion, which appears to be bigger even on treatment compared to the time before. And so you've got this very heterogeneous response in the mother and this very sort of homogeneous response in the infant. And so we think that this is consistent with our idea that there's this sort of very homogeneous disease going into the infant. And so we've got some sort of model of branching evolution that looks like this, where you have these many different distinct subclones that existed within each tumor in the mother, and one small population of cells, almost like a founder effect, left from the mother tumor, and then begin to progress in the infant. And we're just starting to see the early stages of this kind of new subclones arising within the infant. And so that's kind of the end of that work, and it's a depressing but an interesting case of tumor evolution that really allows us to look at some of these processes in ways that we couldn't normally. So I just want to acknowledge some of the people here. So in my lab, Austin Gutierrez he's an undergrad here in the biomedical informatics program who does research in my lab. He did a lot of the sort of single-cell RNA sequencing processing work. Some guys from Jonathan Keith's labs helped out a lot with the single-cell data processing, or the actual wet lab work for the single-cell RNA-seq. Rebecca Halprin, Sen Peng, and Jessica Aldrich all did most of the computational analysis of the mother-infant project, the Transplacental project. And then we've got many other collaborators, both at Mayo and other places, who've assisted, particularly with the Transplacental tumor project. And then as a shameless plug here, I am looking for postdocs or students who are interested in working in my lab. So if you guys are interested or know people who are interested, please pass it along. And that's it.
All right, so I think we have time for a couple questions here, but there's also tea afterwards and lunch. So if there's any burning questions, we can ask them right now. Otherwise, I'll add that students can be at ASU, both undergraduate and graduate students, and be co-advised with TGen. So you can keep your ASU affiliation and get credit for Barrett Honors College, or even be in one of the PhD programs here and work at TGen. So if you have questions about that, let me know. And with that, let's thank Dr. Banovich. Oh,
Carla?
Yeah. I
was holding back.
ask you to wait till the tea then. All right.
Cool. Thanks.
Thank you very much.